We're looking at a technology that honestly offers some massive, world-changing agricultural benefits, but it also carries with it some genuinely severe existential risks. It is the ultimate double-edged sword of modern science, and we're going to break the whole thing down.
Today, we're covering 5 key areas. First up, the promise of genetic engineering. Second, overcoming biotic and abiotic stress. Third, improving quality and yield. Fourth, the existential risks. And finally, safety and mitigation.
So, let's start with Section 1, the promise of genetic engineering, reimagining agriculture.
Okay, let's just dive right into this immense promise and see exactly how this technology could completely transform our global food supply. Because through genetic engineering and modern gene editing tools, scientists aren't just farming anymore, right? They're basically creating customized supercrops. We're talking about engineering plants with delayed fruit ripening so they can easily survive long transit times, frost resistance to withstand sudden climate shifts, and the absolute mind-blowing ability to tolerate complete submergence in water. We can even create crops with photoperiod insensitivity, which just means they aren't restricted by the length of daylight to actually grow. It's literally a total reimagination of what a plant can do.
Moving right along to Section 2, overcoming biotic and abiotic stress, building resilient crops.
But how exactly do these specific modifications protect our food from the pretty harsh realities of nature? Well, to really grasp this, we've got to divide agricultural threats into 2 distinct categories, just like the lecture outlines. On one side, we have biotic stress. Think of this as your living biological threats. Insects, fungi, bacteria, viruses, nematodes, and parasites that are actively attacking the plant. And then on the flip side, we have abiotic stress. These are the raw environmental threats, stuff like highly acidic soil, severe droughts, high salt content, and catastrophic floods.
Now, targeted genetic engineering tackles those living, biotic threats head-on. Scientists have actually successfully engineered insect resistance directly into global staples like maize, rice, and wheat. But it goes way farther than that. We're now shielding delicate crops like chili and lettuce from fungal destruction and equipping staples like soybeans to fight off bacterial invasions. We're essentially giving these plants their own built-in, customized immune systems.
And when we confront the non-living, abiotic threats, the modifications are just as impressive. I mean, genetic engineering has given rice the remarkable ability to tolerate acid soils, survive high salt environments, and even withstand devastating floods. Meanwhile, maize and rice are both being equipped with absolutely crucial drought tolerance, ensuring that even as our global climates shift and water becomes scarce, these foundational crops can still thrive.
Which brings us to Section 3, improving quality and yield, better food, more of it.
Now, you've got to remember, mere survival against bugs and bad weather isn't the only goal here. Science is also drastically improving the actual nutritional quality of the food we harvest. The source material highlights some fantastic upgrades. We're seeing high-protein grains being developed in wheat to better feed a growing global population. We're getting better cooking quality in rice, improved malting quality for barley, and enhanced oil quality in peanuts and soybeans. So we aren't just growing more food, we're engineering structurally better food.
And crucially, this technology directly boosts overall yields. For those foundational crops we talked about, like rice, soybeans, and tomatoes, genetic engineering ensures maximum food production efficiency from every single acre you plant. Higher yield just means more food per harvest, which is really the ultimate goal of all this crop improvement.
Now, this whole utopian vision of high-yield, invincible, super-nutritious crops is absolutely incredible. But, as Professor Kanal's lecture points out, it does force us to step back for a second and ask a really terrifying question. What are the hidden dangers of playing with DNA?
That brings us to Section 4, the existential risks, the dark side of recombined DNA.
Here is where we have to dramatically pivot to the severe risks of recombinant DNA technology. We often think of a biological disaster starting with some massive explosion in a superlab, but here the threat is actually a surprisingly mundane accident. Something as simple as the improper drainage of lab glassware or minor personnel errors could accidentally unleash completely new, highly dangerous forms of microorganisms out into our world. This kind of accidental spread could literally spark the origin of entirely new types of diseases, posing a profoundly serious problem to global health. Furthermore, there's this fundamental issue of evolutionary disruption. For billions of years, nature provided very distinct biological barriers for the exchange of DNA between prokaryotes, like bacteria, and eukaryotes, like plants and animals. But recombinant DNA technology? It just ignores those borders entirely.
Genetic engineering actively interferes with the natural process of evolution by forcing these completely separate classes of organisms to exchange DNA. We are rewriting the rules of biology at a foundational level, and honestly, the long-term effects of shattering those natural barriers remain a massive unknown.
And perhaps most chillingly, the lecture highlights the threat of biological warfare. There is a very real fear that this exact technology could be weaponized. Instead of engineering crops to resist disease, bad actors could intentionally engineer and release disease-carrying microorganisms, leading to absolute, devastating disaster on a global scale.
So, Section 5, Safety and Mitigation, Keeping the Genie in the Bottle.
With threats this severe, ranging from accidental disease outbreaks to literal biological warfare, how on earth does the scientific community attempt to keep this biological genie safely in the bottle? Well, the dangers can really only be minimized through two strictly enforced vital steps. Step 1 is continuously increasing our overall experience and knowledge of the technology so we actually understand the forces we're playing with. And Step 2 is rigorously applying far safer measures to meticulously check and prevent the escape of any new microorganisms from laboratories. Because safety isn't just optional here, it's practically the only thing standing between progress and catastrophe.
As we wrap up this explainer, we're basically left staring at a really profound balancing act. On one side, we have these life-saving agricultural miracles, crops that can survive floods, ignore pests, and feed billions with enhanced nutrition. But on the other side, we face catastrophic biological risks, the accidental spread of new diseases, the disruption of natural evolution, and the looming specter of biological warfare. We have to carefully weigh these two realities against each other, leaving us with one final, provocative question. Are these agricultural miracles truly worth the existential biological risks, and is this really a price humanity is willing to pay? Thanks for listening.